Hello, my name is Karla Paz and my work team is the professors Dr. Yuri Okolotko and Fernando Cobo. My research collaborators are Jose Ortiz and Rebecca Martinez.
This presentation shows the advancement on results of the Agriculture and Environmental Science PhD thesis at Santiago de Compostela University, Spain. It is based on one of the ocean's stressors, such as the harmful algal blooms, that causes the most problems in the marine coastal zone in the Pacific of Guatemala. Everything within the framework of the project strengthening capacities in marine and coastal environments through nuclear and isotopics techniques, through which a coastal monitoring system is being implemented to identify and quantify microalgae. Previous plankton studies in Guatemala are related to algal blooms due to poisoning problems occurred in August 1987, where 193 intoxication of people due to seafood consumption were reported on the Pacific coast of Guatemala. 22 of those cases were deadly. Pyridinium bahamense variation compression was identified as the organism that caused the intoxications. This microalgae produces the paralyzing toxin. Another algal bloom occurred in November 2019 due to the same microalgae. The periodicity of occurrence of this type of natural marine phenomenon is becoming more and more frequent because an anthropogenic impact, climate change, and the study area is circumscribed between major rivers. On the left, Dr. Yasuo Fukuyo, who helped at the beginning of the studies on algal blooms in Guatemala because of his visit in 1995 Guatemala and SEMA on the University San Carlos de Guatemala started the research and studies on the matter. The objectives are focused on the study of the diversity, abundance, and distribution of the community of phytoplankton on the Central Pacific of Guatemala. The research work is carried out in the Central Pacific of Guatemala in Puerto Quetzal as the main monitoring point, where 3 sampling points were established. These were selected based on the red tide monitoring that SEMA has carried out in previous years, having signed with different characteristics. The stations were established in countercurrent direction toward the entrance to Puerto Quetzal, which is the area that communicates with the open sea. At each of the 3 sites, they were sampled at to the depth 1.5 meters and 5 meters. The sampling point, despite being observed very closely, are located with different conditions in relation to the topography of the area. Both are close to the sediment plumes contributed by rivers Achihuate and Maria Linda. Another interesting fact is that if we look at these photographs, we are working precisely in the San Jose Canyon. This is a deep underwater canyon from 200 to 2,000 meters approximately that cuts perpendicularly this continental slope at the height of the 91 angle west meridian and runs into the Central American or Mesoamerican trench that is formed thanks to the subduction zone of the Cocos Plate below the Caribbean Plate. The Mesoamerican trench reaches depths up to 6,000 meters and has steep slopes. These environments are not very stable due to the high levels of seismic activity. However, they are very stable in terms of temperature, salinity, and oxygen concentration. Surface samples are carried out and the thickness of the photic zone is measured by means of an Secchi disk to establish the collection levels in the water column, considering light penetration as the determining factor. Water samplings are carried out with a bottle bandor of 6.4 liters. A phytoplankton net with a mesh size of 23 microns is used. Samples are fixed with lugol acid and stored in Kimax Kimball 500 milliliters glass bottles. At each sampling point, a horizontal drag of plankton is carried out for 5 minutes with a 20 microns mesh size. Environmental temperature and water transparency are measured by visibility of the Secchi disk, water temperature, dissolved oxygen, conductivity, and salinity with a multiparameter probe. In the laboratory, we analyze by the absorbance method total dissolved solids, phosphorus, ammonium, total nitrogen, sulfates, and chlorophyll A using prepared solutions and calibration curves based on the standard method. Phytoplankton abundance estimates are made by means of an inverted microscope, and counts are made in 50 milliliter cylinders and sedimentation chambers according to the Utermol technique. The results show 71% dinoflagellate as the main group in the abundance of organisms identified during the months of January to May 2021. The 6% representing other groups are made up of the following. Cyanophyta represents 46% of the samples with the presence of Trichodesmium erythraeum with an average diversity index of 16.67 organisms samples in the 3 points evaluated in Puerto Quetzal. These species in the months of January and February represented a high percentage of organisms in the horizontal thrones. The dinoflagellate and the identify at an order level. The order mainly found is Gymnodiniales at 56%, representing an average of 207.72 organisms per milliliters, and is made up of the species Acawisio sanguinea, vegetative cells, Marcalephyrinum polycricoides, and polycricoides cyst. The main identified species of the order Thalassiocirales are Laudaria annulata, Planctoniella sol, Skeletonema tropicum. It was evaluated if there were differences in the abundance of species in months and between places. For both cases, non-parametric statistics were used. A Kruskal-Wallis analysis was used. The value of P equals 0.6742, a value greater than 0.05. That means there are no difference between sites, 3 voids and 2 depths in each void. In relation to the abundance of species, the 6 sampling sites have the same abundance statistically speaking. There is a difference between months. The value of P equals 0.004. The abundance if it has variations between months of sampling, not so between sites. There are temporal differences and not in the sampling area. Single statistics, the results show that in 5 months there are no significant differences. For the richness between site, normality exists. A parametric analysis of variance was worked in the one-way ANOVA analysis that showed that there are no differences in the places. With respect to richness, the value of P equals 0.3056. Statistically, all the sampled sites have the same wealth. Based on the species and their abundance, the dendrogram showed that the sites have a similar species composition, although statistically within these 5 months evaluated they are the same. The abundance of the species influence as follows. The organisms were concentrated in the sampling station with the highest densities of organisms, followed by a cluster in the descent order of abundance but representing high species richness. The number of species accumulated in the sampling effort adds up to 111, with 23 points evaluated to date. It can be seen that a defined asymptote has not yet been reached. The individuals computed have 8,784. As of October 2019, sampling began due to the presence of blooms mainly in the months of October and November. These events were sampled in the Boy, Entre Morros and Recalada sector. The collected samples were analyzed with an optical microscope for the total count of the organisms present by means of sodium rafter counting chambers. Regarding the abundance of species and richness, there are no significant statistical difference in the sample stations, but there is a difference in the months of collections. The algal blooms in the Central Pacific Guatemala are subject to river discharge in the rainy seasons, which due to climate change and the presence of La Niña phenomenon this year have increased. It is possible that the increase in previously unidentified species in the Pacific of Guatemala is due to the fact that they are transported in the ballast of the boats, which we hope to verify at the end of this study. Thanks to the ISSHA and SCAR for supporting the registration fee to attend this conference. For more information or if there are doubts of the project, feel free to contact me for any doubts or commentaries.